You can hear me, you can see my slides, and you can see them move.
Yep, everything's good.
All right, perfect. So welcome everybody. Thank you for joining. I am Kurt Veely. I'm the Director of Research at Berry Consultants. This is the second in our webinar series talking about a number of adaptive design concepts and implementation of them in FACS. Today is Group Sequential Designs. I should also mention while I'm here, in addition to these webinars, we also post a fair amount of content on Twitter and LinkedIn. A lot of that content refers back to our website. But if you want to follow me on either one of these, we tend to post things about once a week on various topics and certainly take requests. The outline for today, I want to talk about four things. Generally in this webinar series, we've been trying to divide things up into kind of these four categories. I want to talk about the basics of group sequentials. I know a lot of people know this. Some people might be new to it. I'm hoping if you're new to it, you get stuff out of it, and if you're old to it, you find some of the graphs novel and interesting as well. But essentially, what problem are we solving? Why would we ever want to do a group sequential? How do they work, both from a success and futility standpoint? And what advantages might actually occur in practice? So how much savings do you get? How efficient are they? We're going to talk about the FACS implementation. As with other items in this webinar, we're providing templates. Those are going to be available on our website. Those templates have everything already filled in, but we'll talk about how to change some of the key things that people want to tweak in a group sequential, like interim schedules, what are the futility bounds, and how do I actually see the performance of the design. So we're hoping by providing these templates, people can start with something that's already complete and change a few things and get to where they need to be as quickly as possible. We're also going to talk about some advanced topics. So there's often a discussion about whether bias occurs in group sequential designs. We're going to talk a little bit about that. Talk about delayed outcomes. So, suppose you're in a situation where you don't see a patient's endpoint for 6 months, 1 year, 2 years. How might you still do a group sequential given at any point in the trial? We're going to have a lot of patients that are in follow-up. We don't have their data yet. How do we address that? That'll lead us to Goldilocks trials and the use of longitudinal information. And finally, we'll conclude with some regulatory aspects of these. Does FDA accept group sequentials? This actually is one of the easier things to get accepted from regulators. They're pretty standard, pretty established at this point. But we'll talk about issues that do come up in review, like the importance of the first interim timing, and whether you have information leakage and operational bias, and that will be related to what might you be able to tell from a press release. So diving in, and certainly if you have any questions at any time, Tom is monitoring the chat and will either answer them directly or we'll answer them out loud. So what problem does a group sequential solve? And there are kind of two important settings where you would want to consider a group sequential. Historically, group sequentials have been recommended to save sample size. So I want a smaller trial. And this idea is based on the fact that if I do a sample size calculation and I say I need 250 subjects, well, I certainly know that random data doesn't accumulate information on some deterministic schedule. So if I need 250 to get 90% power, that doesn't mean at 249, I don't know the answer yet. And at 250, suddenly the clouds part and I know what's going on. What is happening is if I do a 90% powered trial, what I'm really saying is I'm going to pick a sample size that's so large that when I get to this final analysis, the proverbial clouds have parted prior to that sample size. So there's a 90% chance that I'll get the answer I desire. I'll get the correct answer I desire before that sample size. But often the clouds parted long before. If you do a 90% powered trial, standard 2.5% type 1 error, you power for an effect X. The trial's successful if you observe 0.6X. There's a built-in protection against bad luck. If my observed data is a little bit worse than the truth, I still get a successful trial. So if we don't have that bad luck, odds are the clouds parted earlier, I get convincing evidence earlier. Can I go ahead and stop the trial at the time that it makes sense to, rather than this waiting for this insurance policy to kick in? So that's kind of historical. It's certainly valid. I think in practice, we see more of the second reason. A lot of clients will come to us, they have phase 2 data. The phase 2 data comes with an estimate of the treatment effect. And, phase 2s aren't the biggest trials. So, there's a range on this. We think our treatment effect's good, but for example, it might be 0.15, it might be 0.20. Both of those are clinically meaningful effects, but there's a range. The confidence interval could go anywhere, from 0 to something beyond good effects. Enough reason to do the phase 3 trial, but not enough evidence to say, I know what the treatment effect is. Otherwise, we wouldn't need to confirm it. So even with this fairly narrow range of uncertainty, suppose I'm only talking about 0.15 versus 0.20. The sample size calculations for these are really different. So we did an example of this fairly recently. For mu equal 0.2, I'm avoiding, there's a variance floating around. But for mu equal 0.2, I needed 263 subjects. And for mu equal 0.15, I needed 467. Smaller effect, bigger needed in. Those are really different. So, a client looking at this is, one of those, not quite twice the other, but it's getting there. And so this would be a big choice if you were designing a fixed trial. Do I do 263? Well, then I'm well powered for 0.2, but 0.15 is a meaningful effect. I'll still be able, I'll benefit patients. There's a market for my drug, but the power is only going to be 68%. That's a little bit low for me. On the other hand, if I do n equal 467, I'm going to be powered for both of them, well powered for 0.15, extremely well powered for 0.2. But in fact, I'm going to be wasteful for the 0.2. I'm probably, those last 200 subjects are not needed. Maybe even the last 300 subjects might not be needed in practice. So, this could be wasteful. So what group sequentials do in this case is they allow for a flexible sample size. I might be able to look around 263 and look around 467 or places in between as well. And so that will let me adequately have a good trial that flexibly sizes where I need it to go. Okay, so how do I do this? I want to try to look at the data often, and I want to make a decision when the information becomes clear. When I know whether the drug works, I'm eventually going to add futility if I know the drug doesn't work, or at least I know the trial won't be successful. Probably the more precise way of putting it. So I'm going to perform interim analyses. So at pre-specified N1, N2, N3, I'm going to have a third party look at the data. Usually in confirmatory trials, you try to maintain the blind. And so this is done by a third party who would not report back the actual trial results. They just report back either you won, you lost, or you should continue. If the data is sufficiently good at these interim analyses, we'll talk about that definition in a second, then we're going to declare efficacy. Otherwise, we're going to continue to the next interim analysis. So look at the data, stop, or continue to the next one. Then look at the data, stop, or continue to the next one, and so on. We would also put in a maximal sample size where this is just as high as we're going to go. So this allows the trial to stop when the data indicate the question's answered. If you have a large treatment effect, the trial's going to be likely to stop with a smaller sample size. And if the treatment effect is small, or at least we'll say modest, but still meaningful, this trial can be big enough to detect it. So you don't always have to go to those large sample sizes, but they're there if you need them. A complexity in this is, of course, anytime we look at data multiple times, it creates a multiplicity. So we can't just test with a p-value of 0.025 multiple times, or the total probability of our type 1 error will exceed 2.5%. This, of course, just it violates the frequentist paradigm. Regulators require you to have one-sided type 1 error. So we can't use p less than 0.025 multiple times. We have to be a little bit more clever than that. So what's done in a group sequential is we pick our interims, so N1 to N capital K. This is when we look at the data. And we pick a sequence of these alpha K values. And so what happens at the interim is I compare the current p-value to this alpha K. And if I'm smaller than that alpha K, then I'm going to stop for success. If I'm not, I'll continue to the next interim. And so these alpha Ks, they're user selected. So you have control over what you pick, but you do have to obey the type 1 error constraint. So the probability of making any type 1 error, so declaring success at the first interim or the second interim or the third interim or blah, 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 the total probability has to be 2.5%, or of course, whatever else you need for the particular trial at issue. Keep in mind that interim results are correlated. So there's interesting math in here. Obviously, the first N2 observations contain the first N1 observations. So you get this nice joint distribution that has correlations based on the overlap in sample sizes. And so because of that, the event's probability stop at the first interim and the event probability stop at the second interim, those become correlated. And so your alpha K values, it's not like these sum to 2.5%, but taking into account the correlation, this creates an interesting math problem of how do I get this constraint to be satisfied? Okay. So a lot of times in practice, what we talk about is instead of directly picking these alpha K values, what we will do is we will pick the alpha spend. And so we will formulate this problem in terms of, well, what's the probability we win at the first interim given the null? And so let's give this a value A1. And then let's think of another event where what is the probability we win at the second interim? And to be clear, winning at the second interim, you had to get to the second interim. So winning at the second interim implies that you did not win the first and you won at the second. So let's give that a probability A2. And keep this going until finally you get the probability of the win at the final analysis, which of course requires you to get to the final analysis. So you will have had to lose all of the interim analyses prior to that. And so let's give this a value of AK. Now, the advantage of this alpha spin formulation is these are discrete events. So either you win at the first interim, you win at the second interim, winning at the final analysis. These are all, by definition, set up to be disjoint. And so if I want just the probability of winning at the null, this is just going to be the sum of these AK values, and this is what you need to have add up to 0.025. So in this case, we often have discussions of aggressive alpha spending, conservative alpha spending. Aggressive alpha spending would mean that these early A's are fairly large. So if I have a kind of 2.5% type 1 error budget, if I'm spending, 1% of it at an early interim, that's often considered to be aggressive. Whereas if I save it where a lot of my type 1 error is at the end, then that's considered more conservative. And so we could consider when you are when you add these up, what do I pick? So what are the possibilities? Also note that if you're given the interim timings and that alpha spending, you can solve for the alpha K. And a note, I'm not spending a ton of time on the math here, but note that you really only need the information fractions. So if you're designing a trial that has looks at 100, 200, and 300, you're going to be doing the same kind of calculation as if it were 1,000, 2,000, 3,000, or 10, 20, and 30. All that matters is the fractions here. And the reason is the correlation matrix is a function of the percentage of overlap in the interims. All right. Tom, is there a question?
Oh, we have just had one come in, yes. With alpha spend, it seems like the probability of win at the interim, given the null, gets smaller as you go further. Is that correct?
So that depends on how you spin the alpha. If you do it conservatively, so one of the ways that's often done, there's a common choice called O'Brien-Fleming. I'm going to get to in a couple slides why that's often chosen. But O'Brien-Fleming, it deliberately picks these first A values to be quite small and retains a lot of your 2.5% budget for the final analysis. So you really only win at the early interims if the data is very strong. So in that case, you'd actually have lower probabilities of winning at the early interims and a higher probability that the trial goes to the end. You certainly could spend your alpha more aggressively. If I spent, if I have a 2.5% budget and I spend 2.4% of it on the first couple interims and don't save anything till the end, then the probability of winning at the final analysis, one, I get there less, but I also, I don't have enough alpha left over to actually, it's not highly powered. So it really depends on how you pick these values of A.
And we have a second question. Oh, sorry. We have a second question, which it's appropriate now, which is that, do the information fractions have to be equally spaced?
No. So there are, and there's a whole, if you dig into the weeds, there's some terminology on this. So a lot of times when people talk about O'Brien-Fleming, The original papers in this area, they did require equally spaced interims. Now we often talk about O'Brien-like spending, where these early papers have been generalized, and you can pick those information fractions wherever you want. So if you have a trial that goes to, say, a maximum of 1,000, you could do 1,200, or sorry, 100, 200, 300, up to 900. You could also start it at 500 and just do, there's no interims until 500, 600, 700, 800, 900. That's perfectly feasible. You don't even really have to have them to be, they could be haphazard. You could do 500, 800, and 957 for your interims. I don't know why you would do that, but there's nothing in the math that requires you to pick them to be equally spaced. I'll talk about different choices in a few slides. So hopefully we'll also elaborate on this for a moment.
One of the advantages of not being equally spaced and the alpha spending compared to the very original group sequential is that you can adjust things if at the point where you have the interim, the amount of information isn't quite what you were expecting.
Yes, so you do add some flexibility for what actually occurs in practice. How much people use that will, of course, depend on how much those are violated. One of the things that people often ask here is, well, what is the right choice of AK? Should I spend my alpha early? Should I do it late? I've just indicated people do tend to conserve it for later. So let's kind of talk about why people would do that. This is an example trial that we looked at a while back. It had possible analyses at 125, 200, and a maximal sample size of 275. So we just decided for exploration that we would just consider every possible way to pick the AKs. So we know we have a budget of 2.5% type 1 error. So we could budget that as spend nothing at the first interim, nothing at the second, and hold it all to the end. We could have spent nothing at the first, spent 0.1% of our budget at the second, and kept 2.4% till the end. And just we considered every possible way that we could divide this up. This is, of course, a grid, but we tried to get every possible way that we could split this up and get an idea about, well, what is the best here? And so we laid out this grid, and for each sequence, we went back, we solved for what the alpha Ks were, and we found the power and the expected sample size for the trial. And if you look at everything in that grid, you end up with a graph that looks like this. So to orient you here, the power of the trial is on the x-axis. Obviously, we want high power, so we want to be over on this right side. And the expected sample size is on the y-axis. Obviously, we prefer a smaller trial to a larger one. And so we'd like to be on the lower end. So basically, we would like to be down in this corner if possible. Every point on this grid is a way of picking the AKs, a way to spend alpha. And so you can identify a few of the points here. This point is always go to the max. So don't spend anything at the first or second interim, save everything to the end. So really you can't stop at the early interims. This particular trial, it does have the highest attainable power, 91.26% in our example. I haven't given you all the details here, but if you fill in the details, every group sequential, if you did this, you'll get this kind of graph out of it. So 91.26%, highest power possible. The expected N, of course, is 275. You're always going to the max. So this is the highest you can get on expected sample size. There are some really bad choices here. So this one, I don't spend anything at the first interim. I spend 2.4% of my alpha at the second. I spend 0.1% at the max. This is not a good choice. And the reason it's not a good choice is it's dominated by other choices. So there are other possibilities that have both higher power and lower expected sample sizes. So why would I ever pick this point when I had any of these points available which dominated? So there are lots of bad choices on here. Most people who are considering, obviously, for in terms of admissibility, you want to be somewhere along this edge. You want to be using a point that's not dominated by another point. Most people, when you ask them what would be your preference, obviously your mileage may vary. But a lot of people would want to pick somewhere in this corner. And the reasoning behind that most people would use is I want to maintain most of the maximal power. So I don't want to lose a lot of power. I want to be in this area, but I want to try to save a lot of expected N. And you can see early here, we've got the ability to decrease sample size pretty quickly while lowering power. So this cyan dot here is a very special one. This is what you'll often see recommended in software packages, this O'Brien-Fleming spinning. You can also, if you've heard of other things, you've done things like Pocock. Pocock ends up somewhere in this range. It's not on this graph, but it's somewhere in here. So Pocock usually, it saves a bit more sample size, but often comes at a cost of a fair amount of power that sponsors don't want to spend. But remember, there's nothing that says any of these choices are bad. They're not dominated by other options. So if you really value sample size more than you value power, then you'd want to be somewhere in this range. But this is why you see O'Brien-Fleming picked. And this is a common graph. If you did this in other group sequentials, you'd end up with this. This cyan point here, just for comparison, the power is 90.68%. 91.26 was the highest we could get. So we're really close. But our expected sample size is 203. So on average, we're saving 26% of that maximum. And this is pretty typical for a group sequential to save, 20, 30, maybe a little bit more percent of the sample size. Keep in mind that expected N, it's an average. That doesn't mean that all my trials are going to be size 206. Some are 125, some are 200, some are 275. And of course, there's a probability of that. So there's some proportion of trials that go to 125, some to 200, some to 275, and the expected N is an average over all those possibilities. These are easily obtainable in a number of software packages. We sell a package called AdPlan. It's designed to do this. There are free packages in R. This is an example of doing it in R. Library GS Design. There's a new version of GS Design out as well. So this is starting to be dated. This part of it wasn't changed, so it's still reasonable. But I've got the function written here using that. And so you can see how I got my thresholds here for 125, 200, and 275. 275. These are the alpha Ks. So they're not the AKs, but they're the alpha Ks that come out of this. So the needed p-values to win. And again, you can see the p-value at the end is still 0.021 as opposed to 0.025. So O'Brien Fleming, it saves most of the alpha to the end. And going back to our graph, this is supported looking through all the possible choices, unless you value sample size extremely. All right. So, the next question to ask is that's picking the AKs, but I arbitrarily chose 125, 200, and 275. So are there better timings for this? Should I pick different values? The most important thing to think about in a group sequential is when do you put the first interim? And that's for a number of reasons. It's certainly in terms of your efficiency of your design. Obviously, you can't stop before your first interim. So the maximal amount of savings that you're going to get is the first interim. If my maximal sample size is 1,000, if I put my first interim at 900, I don't have the opportunity to save very much. If I put the first interim at 100, well, I can potentially stop at 100. There's a but to that, of course. The other thing that you do need is, in most cases, you often have a sufficient minimal N. You need a certain amount of data for safety, a certain amount of data on secondary endpoints. Often regulators will put a requirement on we need to see at least X subjects for various reasons. And so, of course, the first interim timing can't be before that because you can't stop the trial, go to the FDA and go, oh, I didn't get you as many patients as you required. So that first interim timing is quite important. What you may also find is if you're doing something like O'Brien Fleming, remember it saves the alpha for the later sample sizes. So it actually, in general, it probably isn't going to spend a lot of alpha at the very early sample sizes. So you may find that they don't actually generate a lot of practical benefit. While, it would be nice to stop at 100 when the maximal sample size is 1,000, in practice, you just don't do it very often. And so it may not be valuable. This issue is also going to come up when I get to the bias discussion. Early interims can be prone to some bias issues, and there's reasons not to do it. But the first interim is the real decision point. After you've got the first interim picked, generally speaking, more interims are always statistically valuable. So the more interims I add, I'm going to get higher power. I'm going to get lower expected sample sizes. It is always statistically beneficial to add interims, including looking after every single patient. Now, in practice, when I said it's more valuable, there are diminishing returns. So if I'm talking about a 2 interim trial and I go from 2 to 3 interims, I get a certain amount of benefit. If I'm at 10 interims and I go to 11, the added benefit is often really tiny for doing that. And you have to remember that interims have an operational cost. You have to pay a CRO to collect data. You may have to have a DSMB meeting. Usually you do. All of these things require effort, and there's a price tag attached to them. And so what you often will find is there's a practical cap on the number of interims where their operational cost exceeds their inferential benefit. And so what we often do in design is we spend a lot of time talking about the first interims. We may design the trial with a lot of interims, just trying to see, what's the maximum we can get out of it. But then as we refine the design, we'll start to remove those interims. Maybe we designed it with 7 or 8 interims, but let's try it with 4, let's try it with 2, and see if we get most of the benefit. And we might want to remove those because they're too costly. And we would present that to a client. Let me add here. A lot of times the value of a trial is more than the power and expected sample size, especially to an industry sponsor. You're interested about time to market. If we're working in a pandemic, we want to make sure we try to cure COVID as fast as we can. All of these things are going on. If you have a way to compute the net present value of a trial, valuing time and your patent life and everything else that goes into it, you can make this calculation in a lot of detail and really try to optimize your design. There are packages out there. There are some freeware that will kind of do this. I don't think I want to be careful that I'm not overselling what's coming next, but I don't think the freeware packages are particularly good at it. We do sell a additional package to Facts called Quotes, where you can put in all of this information and it will consume Facts output and it will compute essentially the present value of the trial, which that may also let you decide, do the interims have a sufficient cost? And it's related to where you put the first interim and all of the decisions here. Tom, do you want to add anything about that? If people wanted to look at that, for example?
Yeah, no, thank you for mentioning quotes. Yeah, no, we'd be very happy for folks to have a free evaluation copy of quotes. It looks not just at the expected net present value from a single trial, phase 3. It can do phase 2 plus the consequences of phase 2 and phase 3, including the decisions you make at the end of phase 2, whether that's dose selection, the go-no decision criteria, and that sort of thing. So it's quite a sophisticated package, and takes a little bit of getting used to. Hence, we're very happy to give people quite extended evaluation periods on it.
Looks like we also have a question.
Is it usual to perform an interim analysis for futility and subsequent for efficacy?
Not if I interpret that, it's certainly possible that you may do early interims where you only consider futility, and at later interims you consider success and futility. I haven't talked about futility yet at all, of course, but that's coming on the next slides. But certainly you may want to have earlier interims that only consider futility and save the success stopping for later. If I interpreted that correctly.
That's correct. Yes, thank you.
All right, so let's go through a whole example trial here. So a nice practical example. I'm investigating a novel treatment. I've got a dichotomous endpoint. Response is a good thing here. I want people to respond. Based on history, I anticipate our control rate to be about 30%. And I hope that our novel treatment has a 50% response rate. So that's what I'll use as my alternative, and that's what I'm powering the trial based on. If I did this, if I wanted an 80-ish% powered trial, I could run a fixed 200 sample size trial, 100 per arm, one-sided type 1 error, 2.5%, power 83.3. And I can design this as a group sequential and do the first interim at 100. And so this may be a discussion of what's the minimum required sample size And this actually is also, this is going to be an interesting question when we get to bias. I'm going to talk about this again. But for the moment, we're going to do analyses at 100, 120, 140, 160, 180, 200, and 220. And again, this may be what we use for exploration. And, we may consider as we refine the trial, what if we only did 100, 140, 180, and 220? Do we get basically the same performance and we have fewer interims and less cost? We may consider, what about just 100, 160, 220? We'll think about that when we get to the refinement. But we know inferentially lots of interims are good. So a lot of times we design there, and then once we've optimized, we'll back them off. Notice that I've gone to 220 here. This is often the case where you allow the maximum sample size with a group sequential to be a little bit bigger, and that's to allow yourself to get equivalent power. Here's what this trial looks like with success. So I've got down in the table, you can see the various look timings, sample sizes. This is the O'Brien-Fleming p-values required. That get thresholds function I showed in R is how I got those. In the alternative, so if my alternative hypothesis is true, so 30% in the null, 50% on 30% on control, 50% on treatment. You can see the probability that I win at each of these interims, and you can see the probability that I go all the way to the maximal sample size and I lose the trial is this 14.95. The power here is 85%. So, maybe if I was just trying to maintain equivalent power, maybe I could have just gone to 210. I'm about to also add futility. So I've cleverly, hopefully I've been clever, tried to give myself a little bit of leeway so I can add futility and get back to comparable power. The expected sample size here is 156. So if I run this trial, I've got a little bit extra power, and on average, I save 23, 24% of the sample size in expectation. And obviously, keep in mind, it's an expected value. I could stop at any of these, and you can see the probabilities for each one of them. Now, in practice, you should always add futility rules. Everybody should always be using futility rules. It is the most important adaptation you can put into any trial, both because it has the biggest savings that you can achieve, and a lot of drugs don't work, unfortunately. So you want to protect yourself. You want to protect patients. So if the null were true, so the graph I just showed, this is under the alternative. If the null is true, then 97.5% of the time, I'm going to go all the way to the max and I'm going to lose. Well, I don't want to do that. I want to stop early in that case. If things aren't going well, I'd like to pursue other opportunities, allow the patients to pursue other opportunities. So Barry tends to use predictive probabilities for futility. And what we do here is we'll compute the predictive probability that the trial will win from this point forward. So if that probability is low, then I stop the trial for futility. And so common choices for what low means might be 1, 5, 10, 20%. 5 and 10 are often choices that people would give. And so essentially you're asking, this is a benefit risk calculation. If I run the rest of the trial, suppose I only have a 3% chance of winning. Do you want to invest the remaining 100 patients, or however many it is, for a 3% chance of winning? In a lot of cases, the answer is no. Let's move on to the next drug. Let's let the patients move on to the next drug, and so on. An advantage to predictive probabilities is that they incorporate the uncertainty about the current treatment effect. If you want a frequentist version of this, predictive probabilities are Bayesian. Conditional power is also possible. Essentially, you'd be assuming the treatment effect is known in this case and projecting that forward. And here are some various references on where these things come from. Clinical trials, some JAMA editorials here. All right, so how does predictive probabilities work for futility? Suppose I'm 140 patients into the trial. I've got 70 per arm. I've got 15 out of 70 on control, 21%, 19 out of 70 on treatment, 27%. My current p-value is 0.21. So what's the probability we win this trial? We would, if we compute the probability that we win at 220, the maximal sample size, I need a p-value less than 0.018 at 220. So this is just thinking about what I need. I can back solve for what I need to win. So I need 15% observed effect. I only have a 6% observed effect, and I only have 80 patients to go. So if I need to average out this, I need to get to 15, and I've got to average out the 6. So 140 patients have 6%. I need my future 80% to actually have a 32% effect. So the world needs to turn around a lot. I need to go from 6% to 32. That's not particularly likely to happen. I've got a slide here on computing the predictive probability. I'm going to skip the details. One of the purposes of having software is to do this calculation for you. But what you can see here is if you compute this predictive probability, this is a graph showing what I expect to happen in the future. So this is how many control responses I expect to get. This is how many treatment responses I expect to get. And the area of these rectangles is proportional to their probability. Orange means these are outcomes where I don't win in the future. Green means that I do. You can see I'm overwhelmingly likely to end up with a final result where I lose the trial. And so this has a low predictive probability, and I might want to go ahead and stop. It's about it's in the 5% range. Question, Tom?
Yeah, there's a question to, well, actually, oh, there's another one a bit. Could we put the references to the papers that you had on slide 18 in the chat, or are they coming up later?
They're not coming up later. The slides will be available. Tom, I sent you the slides.
Yep.
So if you'd like to copy them out, feel free to do so.
Okay. Oh, right. Thanks. And there's a question about futility in the non-industry setting.
Yes.
So given the earlier remark that the trial won't be successful, is a better description that the effect isn't there. In the questioner's experience, academic trialists are rather tolerant of unknowable type 2 error rates, e.g. use the estimated effect sizes from observational studies in the power, not minimal clinically important ones. Do you agree that stopping futility is a rather bad trial result for science in that case, since it doesn't generate data for the field to decide whether to abandon research on the treatment or not?
Well, so you could certainly, so I'd answer that in two ways. If one of the goals is to demonstrate that a treatment doesn't work, then you may want to have a futility bound that, I mean, you may want to actually say, I need to have a certain amount of evidence that my treatment effect is small, not just that my trial doesn't work. And I think this is what you're saying, is I may want to actually say that. I still want to avoid an ambiguous result benefits no one. So if I know that I'm going to go to the end of the trial and I'm going to end up with something that's not going to answer the question, then I do want to stop. And I think that applies for industry, it applies for academics. I don't want to end up with an ambiguous result, but there certainly are cases where I feel that demonstrating the drug doesn't work is valuable, in which case, that might be, ironically, a trial success in terms of successfully answering that question. So I think I'm agreeing with you, but I do still, there's always room for futility if I'm going to end up in a gray area or an ambiguous area. If I can't change practice, I don't want to invest patience into not changing practice.
Another question, which is, how is the Z value calculated? Can you use a model such as MMRM or ANCOVA?
Yes, you can certainly. This was just a straight probability test, Z test of the proportions, but certainly if you could add covariance into it, you could make this more complicated. Absolutely.
And lastly, could you just go over that figure once more?
Yes, so this is essentially the Bayesian predictions of this trial forward. So if I'm currently at 15 out of 70 on control, 19 out of 70 on treatment, this is a prediction of what will happen at the final sample size. So I expect I'm starting at 15 control responses. I'm going to enroll more patients. I think I'm going to end up right around 23 control responses with, of course, some uncertainty. I think I'm going to end up right around 30 treatment responses with some uncertainty. And I think I'm going to end up in this fairly ambiguous region where I haven't demonstrated anything. But I essentially what I may want to say is if I'm going to get an ambiguous result at the end, I may as well just stop and have an ambiguous result now and go answer a question I can answer rather than a question that it doesn't look like I'm going to be able to. All right, so if okay. Sorry. Oh. Going the wrong way.
So there's a question: what if the endpoint is a survival endpoint? Can you generate a similar predictive probability graph?
Yes, it would probably involve what I think are going to be the predicted events rather than control responses. You could do this in terms of estimated hazards on both of them. You do want to be clever about how you display it, but you could certainly make an equivalent kind of graph on where you think you're going to be at the end of the trial and compute a predictive probability.
I think the prediction is just gets a little bit more labor or computer intensive because you've got to assume you're going to simulate over different accrual and different event rates.
Yep. So I'm going to add a rule to my trial that I'm going to stop the trial for futility if that probability is less than 5%. And so I'm trying to manage a trade-off here. If I do aggressive futility stopping, I allow myself to save sample size, particularly in the null. But I can lose power in the alternative. So we often simulate a range. We discuss with the client. This gets back to finding net present value of the trial. Clients often have utility that depends on what kind of company they are. A big pharma may have lots of other drugs in the pipeline. They're willing to move on to the next one. Someone who, this is our only drug, it may be more valuable for them to give it as much of a shot as they can. Anyway, but just keep in mind, if you're a consultant, you're going to end up with different client needs depending on whether they're an academic, a small company, a big company, so on. So you want to make sure that those trade-offs are understood with respect to the client's values. Or your values if you're doing it yourself, of course, you can be your own client. So I think I talked about all this. Skip that. This is the operating characteristics of this trial. So under the alternative, where I have a 20% treatment effect, you can now see my probability of winning and my probability of losing at each of the interims. The power of this trial is 83.3%, which I conveniently picked my sample size so that I would get back to the, I did a little bit of trial and error to make this thing, come out the way I wanted. But my power is exactly equal to that fixed trial. The expected sample size is 150, and really only 10% of them reach this maximal sample size. Under the null, with a 0% treatment effect, my type 1 error is now, it's controlled at 2.5%. It's actually a little bit lower, given I've got futility stopping, but we'll just talk about that it is 2.5. My expected sample size is 123. And 80% of my trials stop at or before n equal 140. So you can see a lot of the trials are stopping at these early interims for futility. All right, let me talk about facts. And so let me switch gears here. Talk about how to implement this. Share my screen here.
Could you just comment on what the futility boundary was you were using on that for that last
5%,
slide?
5% on that one.
Thank you.
If you use 20, you would get a really low sample size in the null, but you'd lose a lot of power. If you picked 1%, you don't cost yourself any power, but the expected sample size under the null, you don't get that nice result of 80% of the trials stop at the first few interims.
And presumably it's kind of an O'Brien-Fleming kind of shaped boundary, but the other way up.
For futility, not the futility doesn't end up matching O'Brien-Fleming. The success bounds were O'Brien-Fleming, but the futility looks a little different.
Okay. It's, of course, the futility now being driven by the predicted probability, so it's not
right.
It doesn't. Yes, so it's not a group sequential boundary as such. It's just similar to.
So we're providing a template here for this group sequential, where everything's filled in. The hope is that, if you want to change some things, you don't have to, every box, you can kind of have a look. You can see things like the maximal sample size. I want to evaluate how well the trial works at the null, where they're both 30%, how well it works in the alternative, 30 versus 50. I'm basing my results just on p-values. So I've got that all set up. I'm basing futility on the probability of being successful at the trial maximum. So this is the predictive probability of trial success. My design here is fairly straightforward. What I've got, I'm allocating 1-to-1. I've set up my interim schedule, 100, 120, 140, 160, 180, 200, 220. I've also got all of my interims programmed in. So you can see, for example, my futility. If my predictive probability is less than 5%, I stop for futility. If my p-value is less than 0.0023, I stop for success. And you can see as I go through the interims, I'm still 5% for futility in all of these, but you can see the different p-values changing as I go through the interims: 0.0069, 0.097, 0.134, and at the final 0.018. At the final analysis, I'm not predicting anymore. If I can't win at the final, I just don't win. So the simulation here, FACS provides you most of the key information up front, and the rest of it's in the graphs and output files. You can see the average number of subjects under the null and the alternative. These are very close to what's in the slides. I computed those numbers exactly. These are by simulation, but they're quite close. You can see the probability of success, either early or late. These add up to about 0.025 in the null, and they add up to the 83.3% in the alternative. You can see how often we stop early for futility. So in the null, we stop early for futility 95% of the time. If you want to see granularly how that is at each interim, FACS will show that to you. So you've got a time course for futility stopping. You can do this over time or sample size. So again, you can see, most of the stopping, this first interim, 60%, and I pick up another little less than 10 at the subsequent interims as I go forward. But most of the trials are stopped before they get to the max. This gets back at the earlier question. It may be possible to stop, to add a futility interim earlier than 100. Maybe it's valuable to try to get some of these to go out at 80 or earlier if I know that the drug doesn't look like it's going to work. That might be something that's worth exploring. You may not have the option for success stopping. Similarly, for success stopping, if you want to see the entire graph here, you can see the probabilities of this stopping, 20% at the first interim and about 10% at each subsequent interim as you go forward. So our goal, yeah. Go ahead,
just a question. Is this template looks pretty useful? Is that in FACS somewhere?
The template's not in FACS. I was intending that we distribute this as part of with the slides.
Okay, great.
Yeah. I think if you want us to make more templates, we would be happy to do so. So if you want suggestions on particular designs, Tom, I'm happy to do it. I assume we're happy to provide it.
Yes, absolutely.
So if there's a particular design you want, just let us know. And remember, part of the FACS support is, if you want to email us, call us, we're happy to talk you through all of the boxes here. We're trying to give an idea of capabilities, and with the templates saying, here's a place you can start and you can explore what happens if I change this or that box. So for example, with the template, it would be easy enough for me to change the futility criteria. If you want to see 1%, just change these to 1, update, and rerun the simulations, and you can see what's going on here. A lot of times the real difficulty is getting to the first simulations. All right, so let me get back to, can you see my slides again, Tom?
I can. We've got one more question, which is, does FACS display a graph showing the predictive probability of stopping at the end of the trial?
Yes. Well, so sorry, a graph. Remember, the predictive probability depends on the exact data. So you can find those. FACS produces a lot of output files. And so you can look at the output files, and for every individual simulated trial, you can see what was the data during that trial and what was the predictive probability. If you want to make graphs like for any particular set of data, what's the predictive probability as a function of that data, you could also make that graph from the output files. So that's there, but you would have to load those output files into R. There's a button in FACS that lets you open them in R. So we've tried to make that as easy as possible, but the information is there and you can graph it as you choose. But so yes, but a little more effort to do that part.
And how do we determine what prior to use?
So for the predictive probabilities, usually we would often pick something that's non-informative. But of course, if you have historical data, if you have your own phase 2 results, that would be easy enough to incorporate. And given that it's futility, remember the success part is all p-values and frequentists. But if you're talking about a prior to drive the futility, you can pick what you want and it won't increase your type 1 error. So certainly if you have reason to use an informative prior, you could. But often we just use a weakly or non-informative one. Including here.
Thank you. Okay, one more question. For the simulation you just did, is there a way to see how many times the first simulation, how many times in the simulation the first interim futility is not met, but the second futility is met, etc.?
So what we showed when we did the graphs, in order to stop at the second interim, you had to go past the first. So that incremental increase is that probability. You can also get that in the output files.
Yes, exactly. So that was a cumulative probability. So you can see that that step for the second interim was that probability.
Yeah, and if you wanted anything, odd, like what's the proportion of trials that, given I got through the third interim, what's the probability I get to the end? If that's a relevant question for you, you could dig out the output files and directly just calculate that from the simulations. You just basically go, here are the trials that went past the first interim, and here's the observed proportion that got to the final analysis. So in the graphs, you can do a lot of things. In the output files, you should be able to parse any question that's answerable.
Yep.
All right. Okay, so another thing that comes up, so that's the basics of group sequentials. A couple things that come up here is, are group sequentials biased? So we often hear, oh, you did early stopping. You shouldn't do that because you get biased estimates. And that is our opinion, I think shared by others, is that that's really a nuanced question. Group sequential designs can create biased estimates, but you also, in certain situations, you can trust the early results that come out. Here's a paper on this, again, a JAMA tutorial. One of the ways to think about this is it's always worth back solving what effects are needed to win. So I can just look at these p-values. So these are the p-values I need to win the trial at each of these interim analyses. Suppose I just had a 30% observed control rate, just to try to put an anchor on it. I could do this more generally, but I think this is enough to make the point. What do I need to win the trial? So at the first interim, at 100, I need to get a p-value of 0.0023. That requires me to get 31 out of 50, or an observed treatment rate of 62% in order to win that trial. So I need 30 versus 62% is a win. 30 versus 50%, if those are the observed values, that's not a win yet. Presumably you'd continue and see what happens at the later interims. So you can do that for all of these interim analyses, and you can say, here's what I need on the alternative in order to on the treatment in order to win the trial. Now, the most important thing when you evaluate a group sequential is what's happening at the first interim. The first interim comes up over and over again. The reason is, if you're going to get biases, the first interim requires the most extreme results to win. If we're doing this O'Brien-Fleming, you can get odd situations if you're, doing a very aggressive alpha spin, maybe not, but most cases, the first interim's requiring the most extreme results. The other thing that happens is the later interims are often less prone to bias, because if you were on the path to an extreme result, odds are you would have stopped it earlier. So you don't tend to get as extreme results at 120 because extreme results would have stopped at 100. So let's focus on, is the 100 going to be biased or not? Well, one question to ask is, 30% control, 62% treatment, that's a 32% treatment effect. So is that plausible? And the answer could be yes or no. If, our alternative here, we powered the trial for 30 versus 50. If I don't think that a 32% treatment effect is plausible, then, well, what's going to happen is I shouldn't believe that the 32% is real. I don't think it actually can happen. And so, if I do this, I'm expecting that 32% would be biased. In that case, you may want to consider removing that interim and doing things later. I don't want to publish a paper where I have a point estimate that no one's going to believe. I want to change practice. You can, in theory, do some bias corrections, but the bias corrections, what they're going to do is, if I think the 32% is implausible, I'm going to, for example, as a Bayesian, use a prior and shrink that back. I still find that a little unsatisfying. I am a Bayesian. Some people might not claim I am, but anyway, I would claim I'm a Bayesian. But I'm not that fond of publishing a paper where my inference is driven substantially by my prior. I want the data to be most of the weight of my results. So I don't tend to like to let my prior, this 32%, I don't want to observe 32% and then go, oh, because of my prior, I think it's only 10. That starts to be hard to publish. It becomes a referendum over my beliefs rather than over what the data is saying.
Good. What would you do in those tables if it was a continuous endpoint?
So if it were a continuous endpoint, you could still back solve this. So there's still a needed mean difference in order to win. So that would certainly, you do the same thing. It would just be the back solving is a different calculation, but there's nothing's changed about the qualitative picture here.
But you might have some different values depending on also what the observed variance was.
Yes, yeah, of course. Yeah, it would be situation specific. And so I would recommend this is for this example, not, this is not, it's not, I'm not saying that every first interim requires 62%. It's in this example, we need 62%. So typically, a Bayesian would view this as, suppose the situation, suppose that 32% is plausible. I would probably walk into this if I thought that was plausible. Maybe I consider I have a prior treatment effects from 0 to 40%, all equally likely. If you want to put, a more complex prior on it, that's fine. Assume the control rate is, again, 30% for this. So in this case, a 32% treatment effect, it's in the upper end of my range, but it's a plausible value. If I go through and I get my 32% treatment effect, I can compute my posterior mean. And what I find is I do reduce this point estimate. So the point estimate's 32%. My posterior mean's got to be 30.3. But because the 32% is plausible, I'm not pulling it down very much. So you could argue, oh, maybe there's a little bit of a bias here, but I probably don't want to delay getting this treatment out to patients. If this were COVID, I've got a 9-month trial. Do I want to spend 4 or 5 months deciding whether the treatment effect is 30% or 32%? Probably that difference is immaterial. Go ahead and report the result, and just note that this may be a little bit lower. On the other hand, if I thought that effects from 0 to 20% were equally likely, so that 32% is implausible, then you can see that the Bayesian method, it's saying that I think I'm way too high. My posterior mean will be 17.1 because look, the 32 can't happen. I can only get up to 20. So this would be a large reduction. And so this is the kind of case where I'm not so fond of the 100 interim. And in fact, I may not be fond of, some of these interims. I may decide that's a trial where I do want to go to the max. I may still want to stop for futility. But maybe there are some trials where we don't do success stopping because we don't think we're going to get a plausible answer until we hit some minimal sample size that's close to the maximum. So I would think about this always in terms of, what's the trial going to result in, and is this something that's going to be believable? If it is, typically the biases you get to are small. But certainly there are lots of examples in the literature where people have picked very aggressive stopping at very small sample sizes and ended up in an uninterpretable, implausible, people have criticized this, and that's fair for that. So but that's not a statement that all group sequentials are bad. It's like every other method, use it well. Let's see. I think this is what I said. All right, Tom, we've had a number of questions here, which I think is great. I'm going to not cover the Goldilocks here. It's in the slides, and maybe we could. Anyway, we'll figure out a way to get this material to people elsewhere, another way. Is that acceptable? I'm not giving you a lot of choices because I don't have a lot of time to do it anyway.
Yeah. And in fact, we've got another couple of questions.
Yeah, and I'd rather answer the questions. We will make sure that we get this content to you.
So why group sequential design is not popular? I didn't see much. Maybe why work experience is too short. Is that because it complicates the data analysis later on?
So I actually, I don't know that I'd agree with group sequentials. Groups, there are certain, what? Okay, first I should answer this. I deal with a biased set of the world. So obviously I specialize in adaptive trials. Lots of people come to us looking for adaptive trials. So that's what I see. There certainly are a large group of people who I think there are more academics than in industry who are against group sequentials. Part of it is this bias issue, which I think you need to be aware of, but I think you can address. There certainly are, if you're going to do interim analyses, you have to be able to collect data and get all the, you don't need a locked database, but you do need to have data relatively clean throughout the trial. Obviously garbage in, garbage out. So if you're not, if you don't have expertise in doing adaptive trials and making the all the gears work, then this is going to be difficult for you to implement. But there are lots of groups that know how to do this. There are lots of academics trials that run group sequentials. The FDA accepts these. There are lots of CROs that do this. I think historically, academics also stopping early is a little bit awkward. If you've got a 5-year grant, stopping 3 years into it is not so good for your keeping your lab up and running and your employees going. I think there are NIH is being more creative about addressing this. That's a separate topic. But anyway, I think my read is that this is becoming more common, and I see no reason that trend won't continue, that these will become more and more accepted. These are viewed as very standard in industry and regulatory settings.
Another question, which is, aren't the interim looks pre-specified in the protocol? Can, given the 32% example, the timing of the remaining looks be changed?
These would be pre-specified in the protocol. Generally speaking, in a group sequential, they are not allowed to be changed. You can get clever and think about ways of changing it. I don't know that you actually, I haven't seen a lot of examples where you get a lot of benefit from that, especially if you already have a fair number of interims. I've already got, changing 100, 120, 140, et cetera, to 100, 130, 160, et cetera, isn't going to change your life very much. So I don't know that you get a lot of benefit from it. You also very much have to, anytime you want to get what we'll call cute and start changing these around mid-trial, there are type 1 error implications. So generally speaking, I wouldn't recommend changing things around, but I won't say that it's impossible. Just you're going to run into interpretability, and anyway, people are going to look at you a little bit funny and worry that you're playing games with them, even if you're not.
One question is that one can report a confidence interval from a group sequential approach.
Yes.
So do we need to worry about bias? Do we just
so it depends. If you're a Bayesian in this, and I actually I've got a couple blogs on this. If you go back through, if you're a Bayesian, all of the corrections are done by the prior. So this, sorry, I went the wrong way here. So the correction, where to go? There it is. When I take this 32 to 30.3, you can also take the quantiles of the posterior distribution, and they will also adjust automatically. So the Bayesian, you don't do anything special here. The Bayesian machinery takes care of it automatically. And I'm happy to, email us we'll look at the blog. We'll write more on it as needed. From a frequentist standpoint, there are more explicit ways for confidence intervals. In the adaptive design guidance for the FDA, they recommend the materials in this book. And you can also find, this book is 1999, and there are more recent versions as well that are certainly acceptable. Note here, our regulatory experience here is primarily Bayesian, and that's the way we've done it, and it's been accepted to do it in the Bayesian framework. But if you want to do a purely frequentist way, this is where I would tell you to go. I certainly don't want to claim to be expert in something I'm not.
And just kind of jumping ahead to the bit you were unable to cover, if you had a delayed endpoint, what were you going to suggest? MMRM, Bayesian longitudinal model?
I was going to suggest something called a Goldilocks trial. And so this would start to take explicitly into account the incomplete data, both in terms of trying to do predictions on if I stopped now and did follow-up, would I get to a good place? And in addition, that incomplete data also comes with information. So how do you use the information? That's also included in a Goldilocks trial.
So you'd be using Bayesian longitudinal modeling within the analysis. I'm going to include an example of doing that in the fourth talk in the webinar series. In the seamless phase 2, phase 3, I will include the second stage, the phase 3 being a Goldilocks. So we go with a longitudinal model. So Kurt and I can work on making sure that's covered there.
So let me cover I think I've actually covered a lot of the regulatory. A lot of it came out in the questions. So as I said, Goldilocks, group sequentials, these are all things that are well accepted by regulators. In our future things in the webinar series, there may be, we'll talk more about some details. The only issues that we tend to run into in review is to justify the number of interims. You can do lots of interims. Nothing says, you can't do 5 interims. But often FDA will ask you to show that they're valuable, that there is something scientifically worthwhile about doing them. So that's often that is easy enough to show. You'll often have a discussion of binding versus non-binding futility. Some people, if they have a futility rule, they will try to, in effect, a futility rule will decrease the type 1 error. So they might want to buy that back and be a little more liberal on the success bounds. In theory, the guidance allows you to do it either way. But our experience is that non-binding is pretty strongly preferred by the FDA. So you need to control type 1 error without respect to the futility rule. And then going to go a minute over, you may run into concerns about operational bias. The FDA will want you to have procedures in place to make sure that the results of the trials do not leak onto the anyway. You don't want people to know what's going on in the details of the trial. If you want an example, more kind of a more content on operational bias, this will be in the slides, this link. I did a Twitter thread on this on Pfizer's vaccine trial. And, if people were handicapping this as you go, if Pfizer were to announce, we did the first interim and we didn't win, we're continuing the trial. What does that tell you? I did a Twitter thread on this in some detail. These are a couple of the figures from it. The net result of this is group sequentials are pretty good about not leaking information. So not enough time to go through this in detail. But if this were your prior distribution on the treatment effect, which here is really the vaccine efficacy, you can see if I get through a past interim, if I get past interim 1, these two distributions don't change that much. You do eliminate some of the extreme effects in the tails, but you don't really change. Your posterior mean isn't altered, your probability of eventual success. So anyway, a group sequential is nice as you don't have a whole bunch of people, making all kinds of stock decisions on the basis of every little I continued the trial, which is a nice feature of group sequentials. So let me stop there. Thanks for everybody for continuing. The link to the recording will be sent out, I think tomorrow. Tom, after I talk, if you could clarify that.
Yep, that's true.
Will be available. Okay, great. Slides are going to be available. The facts templates will be available. Please email us anything you'd like to know. If you'd like a demo, free evaluation copy of facts. And certainly we post a lot of free content, blogs, social media. We answer questions. We do take requests. We want to be part of the scientific community. So, this part of I'm the director of research. My goal is essentially outreach, education, that kind of aspect. So please contact me for anything more that you'd like to know. Tom, want to close?
Yes, thank you very much, Kurt. Yes. Thanks very much, everyone. And a special thank you to you, Kurt, for the presentation. But I think you've wrapped up everything very well there. So, yeah.
And I forgot a T in the consultants here. So make sure you email Tom correctly.
Whoops. So, yeah.
I'll fix that before they're posted.
Thank you. So, yeah, thank you very much. And I hope to see some of you next week.